So thanks, Anna. It's great to be able to do this. What I have planned is kind of a hybrid presentation because I'm talking to members within the family, so to speak. And so I want to show you how we're framing the project and trying to advance the goals of the project and engage new partners, funding partners, as well as research partners, and so give you the spiel, so to speak, and then also give you an update on where we are with the project in terms of actually, the brass tacks, and that's either one of those avenues is where we could engage on questions and so forth. So we'll get started then. Next slide, please. So the idea is to frame this as a grand challenge, and the goal of the Earth Biogenome Project is to sequence the DNA of all, what we say here, all of life on Earth in 10 years. In reality, the project is focused on eukaryotes, plants, animals, and single-cell eukaryotic species. The microbes have other very well-organized projects that are in the works, and so we thought we would focus our attention on the eukaryotes. But in terms of the grand framing of the project, we talk about this in general terms. Next slide. And, this is the big picture. We're talking about sort of creating a new biology, a new digital repository of information to be able to address all of the grand challenges facing science and society. And I'll show you some slides that are, sort of high-level advocacy-type slides that substantiate this point. Next slide. This gives in detail the position paper for the project. This was published last year. The members, the authorship list that you see here are members of the initial working group of the Earth Biogenome Project, including Kevin, for example, and myself, and others from a variety of different communities that have been involved. Fringy, very active in this project as well. So that will give you, that's in some ways the more substantive version and more extensive version of what we're talking about this morning, with the difference being an update since that paper was published. Next. So some of the general compelling points for this, the idea that we're in the 6th grade extinction. Next. With a number of endangered species identified so far, of course, many of them insects. A quote from Wanming Yang, who is founding individual chairman of BGI, formerly known as Beijing Genome Institute, BGI in Shenzhen, an early partner in the Earth Biogenome Project. And that's important to mention BGI in this context because it reflects some of the nature of the project, which I'll mention again in a little while, and that is we don't seek to duplicate efforts that are ongoing. So as Anna said, i5k is expected to be a strong member of the Earth Biogenome Project's constellation. And in fact, all the work that has gone on in i5k is part of that. And likewise, BGI has initiated some large projects in the bird genomes together with Eric Jarvis and the Vertebrate Genome Project and a large plant genome initiative. And so all these projects are meant to go on. And in this case, the Earth Biogenome Project's role will be to make sure that everyone knows what projects are going on, so large-scale communication, and very importantly, at least strong discussions about standards across the various organismal communities. I'll talk about this later. What is a reference quality genome, and can we agree upon that standard? The goal then would be so that when we are finished, and I'll talk about what the goal line looks like, there will be a level of standardization that will allow this collection of all of the Earth's eukaryotic genomes to be used to the best extent possible. Next slide. And so this is a startling statistic that, that we all should be aware of. All of the advances in genomics, all the generalizations, all the new principles, all the knowledge that's been derived from the young science of genomics is based on 0.3% of the eukaryotic species genome sequenced to date. Startling and really a motivation, again, for the project. Next. So these are some of the general framings. Next, there's kind of rolling slides here so we can put some of the stuff on. And so, you know, when we're talking to a general audience, we talk a little bit about what genomes are, like funding agencies, for example, foundations, government organizations talk a little bit about genomes, books of life, revealing the past, present, and future, some of the big picture, conservation, biodiversity, revolutionizing biology, and, of course, new benefits for society and human welfare. And that's something that we haven't emphasized enough, kind of the new bioeconomy that one could imagine based on all of the knowledge coming out of the genomes, and that's a perspective that we want to develop further, certainly in light of the current political climate in several countries. Just some examples. I don't need to tell this group all the cool things that one can do with genomes, but these are a few. Next slide. So, you know, that's a diagram to show the tree of life, and the idea, of course, is that with all the Earth species, genomes will be able to have a much better idea of how the tree of life is actually structured. What are the evolutionary relationships? What kind of new insights will there be? And of course, obviously, that's a foundation for all of biology. Next slide. Then we can dive a little bit more deeply, which we won't do today, but we can in some presentations into some of these applied areas to be able to engage particular countries, particular donors who are particularly attracted to one or more of these areas. Next slide. Some examples. Again, some of you know these examples very well. Medicines from nature. These are terrestrial plants. And, of course, we sometimes highlight the Nobel Prize, which, of course, has a nice entomological connection. Next slide. More of the same. Next slide. More of the same. Next slide. And then in another domain, looking at the role of genetic variation in agriculture and plant species as well as animal species. Next slide. A little bit about conservation, biodiversity. And of course, I think many of us on the call know how well genomics, conservation genomics has been already established, and so what the prospects could be with an enhanced genome sequence space. Next. So, okay, so that's sort of the background, the marketing, the advertising, the general outreach to potential funding agencies, private, public, individuals, and so forth. And so I wanted to take the time to have that because i5k is fully part of EBP, and one can make the exact same arguments for an i5k project as one can for the whole EBP project. So this information's out there and known to all of you. Now to shift gears a little bit about the ideas for how the project can be done. The backbone of the project will be a kind of a tree of life approach. Next slide.
More like
Sorry? Oh, okay. So phase one will be to sequence one representative of all the known eukaryotic families. There are about 9,000 estimated to be. These we call for at the reference level. So chromosome-level scaffolding. In today's technology, this usually means hybrid genomes, hybrid meaning calling upon multiple sequencing technology platforms, more sophisticated informatics to integrate the data from multiple platforms, and so to have this as a foundation. And so the goal is, the initial goal is to sequence one representative from every family, phase 1. Next slide. Phase 2 is all the known genera, and I'm sorry to insult your intelligence. We just indicate what family and genera and species means for a general audience, so you see that on the right. An estimated 140 to 200,000 known genera, and then finally phase 3. Next slide. Where it's estimated, and I think some of you are more expert than I am on this, between 1.5, 1.8 to 2 million known species, described species of eukaryotes. And so that is the backbone of the project, to go in this orderly fashion, and I'll talk to you later about how that already is playing out in ways different than we imagined when I get to the update on where we are so far. Next slide. Now, of course, as many of you know, the 1.5 to 2 million known species is just the tip of the iceberg. It is thought that there are as many as 10 million undescribed species. And so a question that has come up is, how might the Earth Biogenome Project address this? So the first comment I'll make is that we have decided at this point that we will declare victory for the project when we get to the all-known species, when they are all sequenced. That would be a monumental effort, I think you would, accomplishment, I think you would agree. And we will declare victory at that point. However, we also have in mind a sequencing strategy that we hope to get started during the life of EBP that can address the dark matter of biology, so to speak, the undescribed species. And that is to couple the tree of life approach with a geographical approach to sequence everything in certain biodiversity hotspots, so a metagenomics approach, if you will. And that can, of course, add a whole new layer onto ecology and an understanding of species interrelationships, as well as population biology if some projects go deeper into sequencing multiple individuals from the same species. Anyways, laying the groundwork for that. And then, of course, doing that sort of sequence first, ask questions later approach can, we fully expect, lead to the discovery of new species. So this is a less developed part of the project. It'll be more opportunistic, situational. It'll, I think, reflect the interests of certain countries in doing so, but can fully contribute to the project, as well as extend it beyond its stated goals. Next slide. So some arguments about feasibility, I'll just summarize them here. They're covered in more detail in the PNAS paper. So this is what we estimate needs to be done, and it can be done with current technology at a cost of about estimated to be $4 billion, which is what the cost of sequencing the first human genome, which composed the Human Genome Project, would cost in today's dollars. So it is a project that is technically and financially not out of the question. It's a moonshot for sure, but it's within the scale of things that we can imagine. Again, using existing technology. And again, I don't need to tell this group that if anything, that estimate will just keep going down as the technology gets better. Next slide. So a couple slides coming up about technology development. So on one hand, we argue that we can do the project with existing technology, and that, I think, is an accurate representation, and that's primarily with respect to sequencing technology. We also are aware of the fact that for some parts of the project, especially the front end, sample collection, sample identification, for those parts of the project, we may well benefit from new technologies. There may be species in ecologically sensitive or politically sensitive locations that might require some entirely new approaches. We're envisioning that this project could spur technology development, drone-based sample acquisition, remote identification using machine learning programs that perhaps are available on someone's smartphone. Again, I'm sure many of us here today are tracking all the technology developments and how some platforms are really democratizing sequencing and making it possible to perhaps sequence in locations other than, fully outfitted sequencing facilities such as we have relied upon so far in the history of genomics. This has a particularly interesting geopolitical implication, and that is, again, as I think many of you know, there are different perspectives on the access to biological material in some countries. For example, it's very difficult to have access to biological material, to take out, I guess, is what I mean by access, in certain countries such as Brazil, such as India, and this can directly impact how we imagine the Earth Biogenome Project being organized. One way that can honor the sensibilities that are expressed in those approaches and still allow the project to go forward and help really develop a more global project is to imagine geographical nodes for the project that are fully equipped to be able to acquire specimens, to identify them, and to sequence them, and then create digital data as the item that's being trafficked across the world rather than specimens. And so some of the new technologies might allow for that to happen more easily than others. So that's something that we're thinking about. One thing that needs to happen is the development of a industry advisory board. And we're starting to talk about what that might look like, what industries, aside from the sequencing technology industry, might be there, and what their role might be in the project. So it has a strong technical as well as I said, sort of geopolitical flavor to it. Next. And this slide really addresses another strong argument for why we're advancing the Earth Biogenome Project. And by that I mean the following. One could say, especially those of us that are attuned to genomics, such as those of us on the call today, eventually all the species are going to have their genome sequenced. We know that's where things are going. What is the value in trying to organize that, even though, as I'll show you, we're trying to organize it in as decentralized a way as makes sense. Nevertheless, what's the goal of naming it and promoting it and so forth? There are several goals. Galvanize the biological community, inspire the next generation of scientists, children thinking about this project, growing up to become scientists. Those are important goals. Organizing the project, standardizing it, as we said, so that at the end of the day, the data will be strongly usable and easily usable across different communities and for different purposes. And then another reason to do so is in this slide, and that is to spur innovation in computing. Genomics is big data, a big data science. This is a paper that myself and some colleagues here at Illinois did to try to explore this. We made a certain set of assumptions, including the Earth Biogenome Project, including the development of human genomics with a large number of human genomes being expected. You can look at the paper and see our assumptions. And we wanted to just ask the question of how big is genomics as a big data science? And as you can see, it's big. And the point of doing this, especially with respect to the Earth BioGenome Project, is that there are no tools currently used in bioinformatics that can scale to the level of the Earth BioGenome Project. Even our most fundamental starting point, BLAST, won't scale when we're talking about thousands, hundreds of thousands, millions of different species genome. So one value of putting the computing community on notice to say, hey, this is coming, it's not happening tomorrow, but we're mobilizing and we want it to happen within the next 10 years, is to get the computing community oriented and thinking and working on new algorithms that will scale, that will allow us to derive the knowledge from comparative genomics using new tools. And these, as I say, these tools just don't exist now. It's not a matter of just tweaking an algorithm. They're going to require a new approach. Next slide. So here's a bit of the project roadmap. The main focus is the phase 1, phase 2, phase 3, which, to remind you, family, genus, species. We also want to work in the species discovery metagenomics approach. And so we roughly have the project organized in this way. GGBN is, I'm sorry, blanking on the acronym. If anyone knows it, they can blurt it out, but it's an organization that Jonathan Coddington is a leader in, involves a lot of invertebrate and insect folks, and involves specimen collection. And there indeed are many specimens that have been collected. The Smithsonian is a repository of specimens collected in liquid nitrogen, I forget the exact number, and other places as well. Just to kind of switch to sort of, in-house talk, we're thinking that this is one of the most difficult parts of the project, and Fringy can tell you that this was the case even for our i5k project. Acquiring the specimens, getting high-quality DNA, high molecular weight DNA, especially for the newer technologies, the third-generation sequencing technologies. So that's something that we have to have a lot of thought given to. Next slide. So here's the way in which we're envisioning the organization. We imagine that this Earth BioGenome Project is meant to coordinate existing activities. There are a number of communities that already are active, including i5k, and the idea is not to duplicate, not to get in their way, but to facilitate communication across these organism communities, again, to be able to discuss and hopefully have uniform standards and be able to benefit from that kind of cross-pollination. The geographical nodes, that's really the newest development, and I'll show you a key development that had occurred in that light, and that is the March Through the Tree of Life project has a kind of a 21st century Star Wars global perspective to it. No walls, let's just, no boundaries, let's just get the species and do the project. In fact, the way the project is taking shape so far is more like a 19th century or 20th century nationalist-oriented project. That is, countries and organizations within countries are saying, hey, I'm interested in EBP. I want to sequence all of our country's species. Or we want to do a project that advances the economic interests of our country. And the project can and is proceeding very nicely in that way. We will have to be thinking about how to better coordinate the Tree of Life stuff, the Tree of Life approach with the national based approach. In other words, just to state the obvious, different countries could have some of the same species and genera, and how do we keep to a Tree of Life approach while allowing countries to do what they want to do, or do we revise that approach? That's a topic. It's a positive development because we're getting units, we're getting organizations to step up to the plate, but it's a slight difference in how we imagined it. So anyways, that's the geographical nodes, scientific communities, and we have a working group, and we're now populating various committees on various perspectives for the project, technical aspects, geopolitical, such as how to deal with the Nagoya Protocol, access and benefit sharing, and I kind of talked a little bit about the political aspects of that before. Next slide. So the process that we have is to invite organizations to sign an MOU. We have a standard form for the MOU. Most participating organizations are able to sign the MOU. A few government organizations find some of the language a little problematic, and so they are enthusiastic partners without signing this, and that's fine. But as you can see, many universities and other agencies from around the globe are signing it. This is an ongoing activity. More and more participating organizations can be part of this, and if you're interested in exploring whether your university or institute or government agency wishes to get involved, you can ask me for an MOU. John Kress. There's three of us that originated the project: Harris Lewin from UC Davis, John Kress from the Smithsonian, and myself. Harris is the chair, and John and I are the co-chair. We're moving towards a more evolving and egalitarian kind of leadership structure where the three of us are not like chairs for life, and so what will this look like going forward? There are discussions on the organizational structure, the org chart, and that's ongoing discussion with the working group. But anyways, my point was, if you would like your institution to become a member of EBP, that's a simple process, and John is handling that part of the operation. Next slide. So we had a soft launch 2 years ago at the Smithsonian, and that's where that quote from Huang Ming came from. And there was a nice article in Science about the project. We had our official launch in November of 2018, as this slide shows. We held it in London at the Sanger Institute, and we held it there because the UK is the first country, and the Wellcome Trust is the first organization to have responded explicitly to the EBP call and step up. And so the Wellcome Trust, the largest private foundation in the UK, announced at this meeting that they are committing to sequence all species of eukaryotes in the UK. And the way they're doing this is by setting up a program at the Sanger Institute. There were, I believe, either 3 or 4 programs at the Sanger Institute, and they are adding an additional one called the Darwin Tree of Life Project. So they have their own name. It's their own project. They have a director, but it is part of EBP in this confederation model. They just hired the first director, and that individual is going to be announced shortly, and that individual will be creating staff and then coordinating projects to do the actual sequencing and analysis. So we're very excited about this first project. Next slide. And some other activities that are relevant. The Vertebrate Genome Project announced the first release of 15 reference genomes. There have been dozens, maybe approaching hundreds of vertebrate genomes, but these are done at the reference quality. In this case, the Vertebrate Genome Project is out kind of ahead of Earth Biogenome Project. They're sequencing at the reference quality level for species. They have the opportunity to do so. They have the funding to do so. The Earth Biogenome Project, the collective response is great, go for it. Eric Jarvis is the current head of the Vertebrate Genome Project. And in fact, the Earth Biogenome Project, the next meeting will be held in conjunction with the Vertebrate Genome Project in August of this year, open to all EBP affiliated organizations, meaning i5k. And we're exploring, just developing, this will just be our second meeting, and we're exploring how to instantiate EBP meetings in concert with the organism community meetings. And something for discussion, I think, would be how to have i5k, especially through the Arthropod Genome Symposium that Sue Brown has done such a superb job leading over the years, and now involving 3 institutions, K-State, Kansas State, Notre Dame, and Illinois, how we might perhaps host an EBP meeting in the future. Next slide. The Sanger Institute, which is the host for the Darwin Project, as I just mentioned, to honor their 25th anniversary. They had a project kind of coincident with Earth Biogenome Project, but really motivated by internal Sanger goals. But they published 25 genomes. Some of you know they had a lovely public outreach, science outreach kind of activity associated with this to nominate the species. And those species, quote, come under the aegis of the Earth Biogenome Project in this sort of general umbrella way. Next slide. So just wrapping it up, these are kinds of the ways that we're framing the project, the digital genome repository of life on Earth. And the next slide, last slide. To foment the next revolution in biology. So that's where we are so far. As I said, kind of a mixed presentation of just high-level stuff, the way we're presenting it to potential funders and other groups, and then I tried, whenever possible, to give you the inside story and the update as befitting an i5k conversation. So happy to take any questions.
So thank you, Gene. I just want to go ahead and give people an opportunity to ask questions now before I talk about the USDA's contribution to this project. And so I'll give a moment here. If anybody wants to speak up, you can go ahead and unmute yourself. And if not, I've got a couple of questions that have come in. One question that came in was that some families are more diverse than others. Some may be considered irrelevant. With some insects are more relevant than others. An agreement on this is bound to be difficult. How and who will mediate those sort of discussions?
Yeah, I think the way those are going to be mediated will be a combination of what organization is funding this and what are their interests. And so we have the general goals of the project, and then we have the specific goals of the funding organization and the scientific organization that's doing the work. Maybe those two are the same, maybe they are different, and so there'll have to be conversations. The same kind of question can be raised for species for which the most important information will be coming at the population level, meaning one will need multiple individuals within the same species. It's the same kind of conversation. So the general answer is there's going to be no rule for that. It cannot be a kind of a heavy-handed, top-down, thou shalt do this. We'll have the guidelines, then we'll have the realities, and we'll work them out. And if it turns out that there are some taxonomic units for which one representative is is relatively meaningless, we'll say, okay, we have one, it's not the most useful, and maybe there'll be certain countries, certain projects for which they will have their own reasons for wanting to go beyond that. And so doing more than the minimum will always be great.
All right, another question. So lots of labs are sitting on lots of genomes from lots of species. So what kind of efforts will there be to avoid overlap and encourage sharing of information on what everyone is working on?
Yeah, we need to do this, and I think the general answer has to be communication across all of the organizations that are involved in EBP. So that's going to be on EBP to really reach out to organizations to be able to explore that. It's going to be tough because you know, for some genomes, it's cheap enough to do them that, you know, people don't need funding for it. They're just bootlegging it in their own labs. They have it, and how do we make sure we know all of that information? So we don't have great answers for that. If people have ideas on how we can communicate, how we can set up, you know, I hate to use a dated analogy, you know, like a Craigslist. Just, you know, how do we get that information? I think it's going to be a constant challenge, but it is an important one to address.
So another question: how much money has been raised thus far for the project?
So I would say the only new money that's been on the table is the UK project. And the estimates of this is a private foundation. The estimates of this are in the multiple hundreds of millions that have been committed for this project. They're putting together an entire program. They've hired a director who will staff this, and then, of course, all the costs of sequencing and analysis. And the director, of course, has to lay out the scope of the project. We don't know what the full scope of the project will be. So that's the first kind of post-EBP announcement of funding, which is substantial, and we're absolutely thrilled about it. There are several other countries that are discussing what they're going to be doing, and we would anticipate some announcements from them. But there's, you know, that's the first one. And then, of course, Anna, you'll be talking about the next one, which is on the table. And again, the exact dollar amount is not something that ARS is talking about, but they are committing to this Ag100 species. That's the second project that has been announced since EBP was announced.
So we've got a couple more here. One person, mostly a comment here, that it would be great to have a registry of the genome sequencing projects in the stage that they're in. And I think that harkens to what you said, Gene, about the fact that more communication is necessary in that regard. The last question I believe I've gotten is that, in this person's humble opinion, the metazoans include the microbial partners. And so how are we going to differentiate, since EBP is really not focused on the microbial aspect?
Yeah, so that's a fuzzy dividing line, and I would say certainly when we certainly for the biodiversity hotspot type sequencing, that line doesn't make sense at all, because if the project there has a strong ecological flavor, we shouldn't and we can't ignore those microbes that are in the species. So that's, I think, something that will just generally happen. And then in terms of the line, I think it depends on how these national and international microbial consortium projects develop. What are they looking like? And importantly, what kind of projects are developing from the Earth BioGenome Project, coming from funding organizations, foundations, and so forth. So if the Darwin Project says, you know, we like that idea of doing all the eukaryotic species, but in fact, we've decided we're going to include microbes, and here are the reasons why, again, EBP will just say great and go on. So the line is not firm, but we thought to at least identify and define the nature of the problem in ways that allow us to have a beginning and a middle and an end, we've decided to simplify it in that way, acknowledging, as the questioner suggests, that it is a fairly arbitrary line and getting more arbitrary by the day.
All right. So do we have any more questions? I just want to put that out there one more time, and if not, I'll move on and discuss the USDA's project that we're getting started.
Anna.
Yes.
Can you hear me? Hi, this is Monica. I have one question, actually. Right. I have an announcement for the Entomological Society meeting this November. A couple of us, Fringy, Sue Brown, and Aaron, probably have proposed, apologies about the background noise here, have proposed a symposium on the Earth Biogenome project, specifically focusing on insects. So we'll know on April 15th if that was accepted, and we'll advertise that at that point, too. My second question was, Jean, you mentioned two things in your talk. First was that one of the goals was to have a digital repository that would, among other things, give access to new genomic-based ecology. So that implies to me a couple of things, that there is an infrastructure for people to access the data, and also that appropriate metadata has been collected at the outset of this project, so people can do this genomic-based ecology where people might not know what species there is. So what thoughts have there been among the Earth Biogenome Coordinating Committee about the infrastructure, like the physical, where the genomes will be housed as opposed to what the genomes are, and what metadata will be collected at the onset of the project so that people can actually access the data at the very end, which is the goal?
Yeah, Monica, great questions. First, that's great to hear about the ESA proposal, and I hope it's funded. And to your questions, we have a subcommittee that's forming that's going to be dealing with all the issues associated with sample acquisition, including metadata. And so they're going to be tackling this problem. It's a rich problem, as you suggest. And so that's one piece. And then secondly, there also will be a subcommittee dealing with the data infrastructure. Where will this be housed? How many places? What's the sustainable model? I don't need to tell you how difficult it is to be looking at sustainability of organism-based databases for genomic information. And so we don't have any answers, but other than to affirm your questions, that those are questions that people will be giving a lot of thought to. And I think we'll be really interested to see how the Darwin project shapes up, because that's a large, mega project, and is going to be creating some new infrastructure. And so what's that going to look like? How does that connect up with existing databases, how to take them to the next level, what are the funding implications, all to be determined.
Great, that's reassuring to hear. Thanks.
All right,
great, well thanks everyone, and if you have any other questions that you wish to follow up with me, please feel free to email me. I'm happy to engage. Thanks for the time, Anna.
Thank you, Jean.
Thank you so much. It's really wonderful to hear that, and I think that you'll start to hear those themes that Jean was presenting, and what I want to talk about today, which is not looking at the Earth Biogenome Project, looking at it as a large wrapper for many smaller efforts, including the contribution through USDA, which we're entitling Ag100Pest. So for arthropods, those sequencing projects are really rooted in the i5k project, so I want to start there. And just for those of you, every once in a while we have some people who aren't from the arthropod community on the call, so I'll just mention that the i5k is the international effort to sequence 5,000 arthropod genomes. And this project was kicked off in 2011 with the goal to sequence and analyze genomes of arthropod species prioritized by the community due to agricultural, medical, or biodiversity interests. And so early on, we decided that a pilot project was really what we needed to kick that off and determine what challenges would lie ahead. So that became the i5k pilot project at the Baylor College of Medicine, which was led by Stephen Richards, or as he's known to our community, Fringy. And so the i5k has since continued as a community of communities, and you can see that by some of the various logos those groups have developed here. So as we stand now, though, the i5k includes several additional efforts under its banner, most of which I won't have time to describe today. But funding for these has come from diverse sources, including the NHGRI Model Organism Program at NIH, which funded the pilot project, the USDA Agricultural Research Service for the Genome and Ag100Pest projects, as well as the i5k database, i5k workspace at the National Agricultural Library, and then the French National Institute for Agricultural Research, or INRA, as well as the University of Memphis and NSF for the beetle project. So I think that what you'll see here is, like Jean said, is that we have to really look to a lot of diverse sources for our funding of these projects. So the i5k workspace at NAL is one of the investments USDA has made. And it's a database that hosts, quote-unquote, orphan genome projects, essentially those that don't necessarily fit the model for other arthropod genome databases, such as vector-based. And so as it stands, the database hosts 70 genomes and is continuing to grow, and supporting data visualization, manual annotation, and search tools, as well as training and outreach efforts to our community. So the next, the USDA has collaborated with the Smithsonian on what we call the Genome project, and the goal is to sequence non-target arthropods in the corn agroecosystem in order to evaluate available pest genomes alongside non-target insect genomes, looking at pesticide resistance in order to improve biocontrol efforts. So given the interest in using RNAi approaches for pest control, we really need to first make sure that the target RNA is not present in beneficials, and that's what this project is geared towards. So really what we want to focus on today here is my agency's latest project, which is the Ag100Pest initiative, to sequence the genomes of the top 100 agricultural pests in the United States. So at this point, this will be our commitment and contribution really to the Earth BioGenome project. And as you'll see here, we want to really focus on high-quality genome assemblies. So the leadership team for this is myself, along with Kevin Hackett and Brian Scheffler, as sort of the executive team. And then we've pulled in a lot of other partners across the USDA Agricultural Research Service to head up specific teams, because we really want to approach this in a very systematic manner. And this has represented a lot of investment from USDA to get this project off the ground, not just looking at creating infrastructure within the USDA for the sequencing side of this project, but also the people infrastructure to bring this together. So we have broken this down into having a prioritization team, an extraction and pre-sequencing team, a sequencing team, an assembly and analysis team, and then a post-assembly team. But I want to get back here first quickly to what I said, is that we want to do high-quality genomes. And so this slide was generated toward the end of October based on some work that I did pulling out at this point, over 366 genomes in NCBI's database. And as you can see, there's great variability in the depth of sequencing and the final assembly contiguity. The contig sizes range, or did at the end of October, from 319 base pairs to 26 megabase pairs, and scaffolds ranged from 457 base pairs to 410 megabase pairs, a really huge range. So given the standard techniques and technology available for many of those previous efforts, such as the i5k pilot, they were really using short reads, and they were able to produce perhaps mediocre to really excellent draft assemblies. And with that pilot, we did really learn about the challenges of sequencing arthropods, and thankfully also a lot about their cool biology and evolution. But with ever-improving sequencing technologies, we really want to move the goalposts and push for higher quality assemblies. So when we talk about reference quality genomes in the context of the Ag100Pest program, we're aiming to meet the EBP standard of 2.3.2 QV40. So this is something that you'll see mentioned if you look at that PNAS article that Jean was discussing about the EBP. And this specific figure is taken from the supplemental of that, and it describes how that score is created. But essentially, when you're looking at long-read technologies, which is what we're hoping to use, as well as scaffolding where possible, and we're hoping to, in this context, use that for maybe our really top priority species, that is the way to meet that. And so if you look at the current arthropod genome quality in NCBI, I've drawn this red line for what the genomes are that would meet at least two of those statistics that you can easily pull the data down from NCBI and assess. And so at this point, or in October, there was only 41 arthropod genomes that would meet that 2.3 standard, which is contigs of 100 kb or better and scaffolds of 1 megabase pair or better. Now, all of those do not meet the MAP status and QV status statistics desired by Earth Biogenomes. However, we expect those metrics will be attainable with long reads and the ability to sequence single specimens from smaller inputs of DNA, such as using the low input protocol that PacBio is releasing. So we know, however, that one size does not fit all, and we are going to have to rely in some situations on pooled insects or other technologies for 10x in certain cases. So this slide sort of shows you that overall project workflow. And as I mentioned, we've set up a lot of teams. And so I've highlighted those with different colors so you can kind of get a sense of what we're aiming for here. Again, we're looking to use long-read sequencing on single individuals where possible. We want to generate RNA-seq data to support both gene prediction and manual annotation efforts. I think often RNA-seq is sort of a forgotten necessity when you really want to focus on having good gene models. So that is a key aspect of this project. We've also invested in a functional annotation pipeline that's going to be tuned towards arthropods. And so that's one of the really unique aspects of Ag100Pest that you won't necessarily see in other EBP projects, perhaps, is that we've invested in this pipeline, and it's a cooperative agreement with the University of Arizona's Fiona McCarthy. And so this pipeline will be an open source pipeline that's going to support the annotation of Go terms and KEGG pathways for gene sets. And one of the other relatively unique aspects of the Ag100Pest is that we really have a database that's already gearing up to host all of these genomes. So we'll have support for data visualization and manual annotation of those gene sets through the i5k workspace genome database. So just to speak a little bit about species selection, at this point we've received around 400 nominations across at least 10 different orders of arthropods. We've specifically sought those nominations from APHIS, the Federal Interagency Committee on Invasive Terrestrial Animals and Pathogens, the Cooperative Agricultural Pest Survey, many USDA researchers, as well as the general i5k community. And we've got diverse nominations across, when we talk about ag pests, we're looking at not just field crops, but also animals, bees, forests, and stored products or stored grain pests. And some of these nominations do include foreign pest species, which are considered high invasive threats to U.S. ag, so that we could perhaps have tools at the ready in case those pests do actually succeed as invasive. So I'll mention here that you see at the top, the spotted lanternfly. And so on last month's webinar, we discussed, this was discussed a little bit because this is a sort of a unique and collaboration between the University of Pennsylvania, it's Penn State, USDA, as well as PacBio. And so one of the neat things about this is that, so let me just tell you a little bit about spotted lanternfly. This is a highly aggressive invasive pest to both grape and hops, which are two obviously treasured commodities, as well as hardwoods. And so this has been a neat collaboration in the sense that this was sequenced by PacBio on their Sequel II system, and it's really opened up our eyes to the growing capacity of the technology, and it's allowed us to really look forward to what we will have available at our disposal. And so we're looking forward to publicly releasing this genome soon, and that's one of our really mastheads for this project at this point. So obviously, though, we have goals beyond just generation of genomes. We really want to look at this as infrastructure for larger research questions, and so we're targeting the species that we're selecting to species that will help us answer bigger questions and really benefit projects that are already ongoing. We're also looking at generating best practices and decision trees for various stages, including DNA extraction, sequencing, design, assembly, and annotation, and those will be released within publications, scripts, and other forms of documentation to the community to really not just, again, generate genomes, but to create a knowledge base around how to do a project like this. Like I've also mentioned, we're really focusing here on capacity and expertise building within the USDA's Agricultural Research Service, and then community building within and outside of our organization as well. So I just want to thank quickly some of the principal investigators of the projects that I've mentioned. Again, Frenji, who previously had been at Baylor College of Medicine, is now at UC Davis. The i5k workspace is headed up by Monica Polchow and Chris Childers. Cornell, which is a large effort mostly headed by Jay Evans and Brad Post, as well as John Coddington at Smithsonian, and then Ag100Pest, which I've already mentioned. But I want to say that all of you are really, truly the investors within this project, and we're looking forward to having your input on how we on collaborating on many of these species in the future. So I'm going to go ahead and leave this slide up, and I want to make sure to open up for questions at this point. And so it looks like we've already got some coming in. So if anybody wants to, feel free just to go ahead and unmute. But I've got one here. Is Ag100Pest membership limited to USDA personnel that you're starting the AgriVectors project? This is Surya. That will fit nicely with the goals and will produce high-quality genome assemblies and web resources for 3 to 10 hemipteran species. So the membership isn't, most of these projects essentially have a champion within the USDA at this point, but we certainly have already been discussing projects that are, as we said, collaboration with outside groups. And so this is one that I want to look at to see that as these are priority species that stakeholders within these ag industries would look at, then those would be also how we would prioritize species. So I think this is an exciting thing to do. It's always better to pool resources, and I think that these sort of collaborations are an amazing way to do that. So let's see. So, Meg, I don't know the answer to your question about the spotted lanternfly. If it's a pest of hops, does it also attack cannabis? I don't know. I suppose that would make it a much more valuable or a much greater threat to some industries and some groups. I wouldn't know the answer, but I know it's a great source of concern in the Northeast right now, and it's already been spreading. So we're really excited to get this genome done and start being able to look better at other control strategies for it. And so is there any other questions that anyone has? We've got the lines open as well as anybody. And yes, somebody else brought up a point. That was the other thing I wanted to say about spotted lanternfly. One of the reasons that we moved quickly and initially with that project is because every agency that I mentioned previously about how we prioritize insects, let's see if I can get to that. All of these groups stated that spotted lanternfly was a priority species, and I think you all understand how rare it is to get consensus on what a problem is and what is a top insect. And this, and I believe there was only one other insect that might have reached that level of multiple nominations at that phase. So, so again, that is really why we prioritized it so heavily. Is there any other questions? Well, if there's no other questions coming in, I'm not seeing any, and I'll leave the line here open for just one more moment. All right. Well, I think I just want to thank everyone again for tuning in. I'm excited about this project. I'm excited about working with the community on this. I think we'll have more to say about the project and some of the other species that we're going to be looking at when the Arthropod Genomes meeting, Arthropod Genome Symposium comes around in June, and I hope to see everyone there. And then again, when the ESA meeting, hopefully that symposium will kick off, and we'll have even more to report at that point. So thank you again, everyone, and stay tuned.
Thank you, Anna.